And so this is the arena of what I call personal genome sequencing. Try and sequence a healthy person's genome, predict what could go wrong, and then manage that state. And so one way of thinking about this is illustrated here, that you would take your genomic DNA. Remember, this is 6 billion bases. It's not trivial. That's 200, well, I was going to say telephone books, but nobody has telephone books anymore. But, well, this crowd's as old as I am, so you know what that means. 200 telephone books. It's a lot of information. And basically what you do is you sort through this for your, you look within your genes, there are about 20,000 genes, and you'll try and predict your disease risk. You'll try and maybe diagnose disease early based on this information. You might monitor a disease state based on this information. If you catch something, you can see whether it's, this disease might manifest itself. Minimally, from pharmacogenomics, you hope to be able to use the information to learn something about treating the disease. And just like the mystery disease, at the very least, also, you would hope to understand disease states if a disease does come along. And you wouldn't do this in a vacuum. You would bring in other sorts of information. You'd bring in your family history. You'd also bring in the medical tests and medical exams you're doing to give you some sense of, together you'd combine all this information to try and make your best predictions. Now, one thing about our lab is our lab does do a lot of this genome sequencing I mentioned, but we do a lot of other large-scale characterization. And specifically, we've invented technologies for looking at all the RNAs. So your DNA makes something called RNA, which makes something called protein. Some of you know this, some of you may not. But the point is that you have 20,000 genes. They actually make at least 100,000 RNAs, and they probably make millions of different proteins. And our lab has been involved in developing technologies for actually measuring all these things and analyzing them over the years. We worked on this for about 20 years. And it always bothered me, in fact, especially recently, that when you go to a doctor's office, they measure 15 things. And you give back this report, right? You guys look up online, you see a few little measurements there, and oh, well, my cholesterol's high, and that's great. And as someone who measures literally tens of thousands of things, I just thought this is ridiculous. Where the field should be going. We should really be measuring large numbers of things. Now, I can't tell you I know exactly which things we should measure. That's why we try and measure everything. And specifically, what we wanted to do was measure all the transcripts, proteins, metabolites. Also, not just 15 things, but try and measure as many things as possible. And so we set up something we call personal omics profiling. And if we integrate all this information, we call it integrated personal omics profile, or IPOP. Okay? And the idea is that in addition to your genome, you'll also measure modifications on your genome. Again, this is a little bit for the aficionado, your epigenome, meaning your DNA can get methylated, and that responds to the environment. And so this is one thing we try and measure. We measure all the RNA that comes from your genes as much as we can. We measure all the proteins. We're doing most of this in your blood because that conforms to the medical establishment, and also because your blood is essentially the garbage can, if you will, of your cell. Everything, it interfaces with all your organs. If something goes wrong, signs show up in your blood and ultimately get passed through and filtered through to your liver and kidneys and things to ultimately get excreted. And so the blood is a good system. That's why people do monitor blood. So we try and measure all the proteins. The technology here is such that, in fact, from some of the things we've invented, you can measure all of your RNA that will be in your blood cells. We can measure about 5,000 proteins that are in your in your blood cells and in your serum, which is the circulating part of the blood. We also measure this measures the most abundant things. We actually dig in and measure these rare things called cytokines that stimulate. They're important for your immune system. So we also make sure we follow those 51 or so of these. We try and follow all the metabolites we can. We follow 5,000 peaks in a mass spectrometer. We use a lot of mass spectrometry for this, although we only know what 1,000 of those peaks are. This is the state of the field. It's still, in many respects, early days. On the other hand, following 1,000 things is much, much better than I guarantee the report you're getting from your doctor. This is still a lot higher level than we've been looking at before. We also follow all the antibodies that are circulating in your blood. We see which ones you have circulating. We think they're a very exquisite monitor of disease states. And we actually set up technology so we can follow the reactivity on proteins. And with the latest technology, we can follow all the reactivity against all your proteins and against all the viruses that are known. Okay, so we can actually follow your antibody profile, if you will, against everything you've been exposed to recently for viruses. We're setting up for other pathogens like other microbes, bacteria and stuff as well. And then most recently, we've been setting up methods to follow your microbiome. You may have heard about this. You actually have more cells in you that aren't you than are you. Meaning we have about 100 trillion cells, but you probably have about 10 times of that are bacteria. Okay? And they're in your gut. They're everywhere. They're mostly in your gut. And so the microbiome we follow is actually the gut microbiome, which means your poop, urine. We follow the tongue. We follow nasal. And we follow skin behind the ear. So we follow 5 different microbiomes to see what kinds of things are going on there. Now this part and this part have only been added recently, and so most of our work has really been on this. So initially when we started this study, we followed about 40,000 different molecules in the blood, and now we literally make billions of measurements in blood, okay, to follow in detail what a health state looks like, what a blood state looks like, okay? Now, when we rolled this out, the story behind this is that, I forgot to say, the way we got involved in this is I was at Yale for 23 and a half years. I moved here in 2009, and we had been talking about doing this kind of project, and when I came here, it was kind of a good excuse to do it. When we decided to roll this project out, we wanted to get material from someone who would be very accessible, because you have a way of running out of material when you do experiments in science. And the other big thing was there was this controversy raging at Stanford at the time where they wanted to let medical students genotype themselves. And there was a group of people who said, this is absolutely terrible because they may learn, it was optional class, but a lot of people were worried that they may learn their father's not their father, or they're at risk for Huntington's or something like this. So, and they were saying, absolutely not. We should not allow this class. They should just get somebody else's genotype, some random genotype means look at some of your variants. And then there's a group of people who said, why not? You can go do this in 23andMe for 99 bucks. And if you do it in this class, we'll teach you more what it learns, and you learn from the world's experts, and it's optional. You don't have to do it. You don't have to take a class. You don't have to do it. Anyway, it's very big controversy, and I stepped right into it when I showed up at Stanford. And so I didn't want to deal with any of that. So when we decided to launch this project, we decided we'd do it on me, because I didn't have to worry about suing myself. I get lots of material. Very safe approach. And we've actually been doing this now for about 3.5 years. We've been taking samples of my blood and running it through these measurements. Initially 40,000 measurements, now billions of measurements. Okay, and so the day we started this was actually right here at this HRV, the 0 time point. It so happens I had a viral infection that day. It's very congested, so we took blood that day, and then we took it 4 days later and 21 days later. And then, I didn't say, but we actually took a sample like any scientist. When I enrolled in a new health plan here at Stanford, of course, I saved the tube of blood and kept that in my freezer. That was actually very valuable. And then every 2 or 3 months, we take another sample, and we do take them at interesting times as well. So when I'm healthy, we take samples, again, every 2 to 3 months. But I have a little kid, so I get sick at a pretty high frequency. It actually turns out twice a winter. So 9 months after this first one, I had an RSV infection. And of course, we were very prepared this time. As soon as I was congested, we took blood that day. You can see the next day, 2 days later, 2 days, 3 days. So we do dense sampling when I have a respiratory virus infection, and then every 2 to 3 months when I'm healthy. And you can see I get about 2 infections per winter. And I've had 3 rhinovirus infections, 2 adenovirus infections, and one of these respiratory syncytial virus infection, which is quite nasty. That's actually unusual for adults, very common in kids. All these are colds, basically, of different forms. And okay, so those are the infections I've had. And so, again, we sampled. There's now about 62 different samples collected during this time. We started out by sequencing my genome. I won't, because I've gotten a little later, I won't spend a whole lot of time on this, but you find all these single nucleotide variants, just like I was telling you, indels and structural variants, so we find all the different changes I have in my DNA. And then we go through and we try and actually understand how these might impact diseases. And we use 2 different approaches for this. One is you look at changes right within your genes. And I won't go through the details of this, but if this is your whole list of variants, we tend to look within the genes. And not because that's necessarily where all the important changes are, but that's where we can have our best shot at interpreting what might go wrong. So we look at changes within the genes. We look at those that actually lead to changes in the protein. And we look at those that, by computationally, they're predicted to actually damage the protein. Okay, so we go through, we look at the changes, look for those that might be damaging for the protein. And believe it or not, there's about 4,000 genes in a database that have been associated with human disease. So you zoom in on those 4,000. And so basically, we're looking for changes, again, that will be in disease-associated genes that are predicted to be damaging. Okay? And then I didn't explain, but we also look for those that are thought to be not common, because the thought is if it's common, I'd know about it already, and it would be known, although that's not 100% true. So I didn't say, but this pipeline we make makes lots of assumptions, all of which are wrong, but it's the best we can do right now for interpreting this kind of change. So what you'll notice is from my 3.5 million or so variants changes, I have 51 that are damaging and associated with human disease. And in fact, this is my list of damaging changes, okay? So lesson number one is, if you're a worrier, you should not get your genome sequenced, okay? Because you will have a list just like this. In fact, you'll have, especially if you're in the same ethnic group as me, which I can see many of you are, you will have some of the same variants. Not all, in fact, probably a minority, but still some. And the point is that we all have this many changes. We all have lots of changes. Because we've zoomed in on those that are rare, it's usually one of your two copies that's changed, not both. Although one copy in many cases can be damaging. When we looked at my list, that actually was fairly benign overall, believe it or not, in spite of what I just showed you. Okay? But there were three things that caught our eye. One is I have a change in a gene that's called TERT, which is it's a gene in a protein called telomerase, which adds sequences onto the ends of your chromosome. Now, why is this important? I have the exact same mutation as people have aplastic anemia, so a blood problem. And I don't have aplastic anemia, but that certainly did raise my eyebrows. And I have the exact same mutation. They have one copy mutated just like me. Okay, so if you read the literature, in fact, you would assume I should have the disease. Okay, so we did measure the ends of my chromosomes. They're actually a little bit shorter for my age group. Your chromosomes all get a little bit shorter over your lifespan, by the way. Okay, mine were a little bit shorter, about 9% for my age group, but so, you know, they're still plenty long enough. After seeing that, I was not so worried. And also, we went out and sequenced my mom because we actually wanted to assign variants onto her chromosome. And my dad had passed away, so we couldn't do that. But we could assign which changes belong to which chromosome. That's important for a lot of the interpretation. And it turns out she has the exact same mutation as me. She's 85, no aplastic anemia, doing great. Okay, so the bottom line is we think that this change is contextual, meaning it's a problem in certain people but not in others. And this is one of the challenges in interpreting these genomes. You don't always know, but by collecting a larger database, we hope to. So ultimately, when there's millions of people getting their genome sequenced, we'll be able to interpret this better. Okay, I did think I was in the clear once I sequenced my mom, although I figured I'd probably be measuring the ends of my chromosome for a while. But then I saw someone give a talk recently, and there's another lesson, which he was measuring the ends of chromosomes for people of all different age groups, thousands of people. And as I say, your chromosomes get a little bit shorter over your life. Well, it turns out for women, that's a very narrow slope. They just get a little bit shorter. For men, they start out going a little bit slow, but at age 50, then it hockey sticks down. So even though my mom is in the clear, I may not be in the clear. I'm 57 now, actually 58 pretty soon. So in fact, I may have to worry, but, well, not too worried, but if I do start looking pale, that's what anemia means, then I'll probably look a little harder at what's going on here. So anyway, this is the kind of thing. So the point I'm making is that you actually can use this information later if problems do arise, okay? I hope they don't, but at least I have the information. Now, other things that have popped up, I knew I had a high cholesterol going in this study. I'm a great responder to statins, Lipitor initially, now I use simvastatin. And I'm a good responder, and almost certainly I have two different changes, one of my mom's DNA and one of my dad's that are slightly different, but they're both inactivating. They're killing that gene. That's most likely the reason I have high cholesterol. And I also had some changes that suggested I might be at risk for type 2 diabetes, but they were just a few we spotted initially. We did another kind of analysis that's very similar to 23andMe, where instead of just looking at these single changes that are likely to cause disease. Again, this is getting a little bit technical, but we can actually look at common diseases, and there the effects are thought to be small. So you look at all the little they're changes that lead to little effects, but you can add them up. They're called risk alleles or protective alleles. This is what 23andMe does. So you look at all of these different changes and you add them up. And the tool Butte has set up an awesome database that actually has collected all this information, and then you can predict risk. Okay? And so this is what they call a riskogram, and you get something similar to this from 23andMe. Okay, and as a European male, this is a 100% chance of getting one of these. There are hundreds of diseases, by the way. And there's a 0% chance over here. And the triangle is my starting point for getting one of these. And then from my DNA, I can predict whether I'm either increased risk, which is the orange bar going to the right, or decreased risk, which is the blue bar going to the left. And for the most part, this made sense with my family history, which was, I guess, scientifically reassuring. That is to say, here's my high lipids. My high cholesterol is the same as high lipids. You can see I'm in increased risk here, and it shows up in another spot here. That's a tiny blip. It actually goes out a little bit further of higher coronary artery disease. Everyone on my father's side has died of heart failure, so that made a lot of sense too. Low obesity. Nobody in my family is ever overweight as far going back to my great-grandparents, so that all made sense. But there were some surprises, and there were two that really caught my eye. One's fairly recent. This we discovered it sort of rather later when this information came along, but I'm at very high risk for basal carcinoma. I was not aware of that in my family, but we had a family reunion last August where, of course, we bled all my brothers and sisters. But I happened to ask them at the same time if they knew anything about basal cell carcinoma. It turns out two brothers and one sister each had several basal cell carcinomas, and they had them removed. And so the point is, it was in my family history. I just didn't know it. It's not the kind of thing you talk about around the coffee table or the dinner table. So the net result is I would argue that your genome is a better measure of your family history than your family is, because your family is all reporting it, unless you've got it somehow better written down than I do. Okay? So that's one. But the other thing that popped up very early on, you may have noticed, and that's the story that Marty introduced, was this increased risk for type 2 diabetes. I went from about a 25% to 47% chance. And that was perplexing because I wasn't aware of that running in the family. I talked to my mom. You'd be amazed when you get your genome sequence, you will talk to your parents quite a bit about what's in your family, this sort of thing. And so my mom basically said, well, your grandfather lived to be 87. He got diabetes late in life, he cut off sugars, he got about 67 or so. And so that's why I'd never really heard of it, because it didn't seem that big deal. And I have like 13 aunts and uncles, and so plenty of grandparents. So the point is, 1 out of whatever that is, 17, didn't seem like that big of a deal, and that's why I wasn't aware of it. But remember, we were doing all this omics profiling that I told you about. We were measuring the metabolome and the proteome, and amongst the things we were measuring was glucose. We did all the standard medical tests as well. And it so happens that my sugar was running along just fine, quite normal, in fact. This is a very normal range. It doesn't matter whether it's fasting or not. Normally, the best measurements are after you're fasting. But what you'll notice is running along fine, and then it so happens it went up. And ironically, the day it went up was when I went in to get a glucose metabolism test at Stanford, because we had the world's experts there. And I went in to get this test, and the woman looks at me and says, well, it's unlikely you would have a glucose problem. You don't have a family history. You don't look like you should have a diabetes problem. And I said, yeah, but I sequenced my genome, and I wanted to see if there's anything wrong here. And I think to humor me, she started this. But even before she did the test, basically it came out high, 127. It's pretty high. We're both a little surprised. So she repeats it, comes out the exact same number. And she went on and did the test, and I'm basically insulin sensitive, which is fine. That's associated with diabetes if you're insulin resistant. But the point is, because it was high, I went in, did a test a week later, and basically it came out even higher, 140, and there's a better measure of glucose levels called hemoglobin A1C, and this is actually borderline diabetic. 6.5 is officially classified as diabetic. Now, I didn't know anything about type 2 diabetes because it wasn't running in my family, so I figured I'd better go to my general practitioner, which I did. And I'm a busy guy, so it took a month to get there. And when we measured, she looked at me and said, there's no way you have type 2 diabetes, you don't look it, da-da-da. So But then she did, she pulls the blood and sends it off to test. Probably sent the exact same place the first ones did. But anyway, it came out higher. You can see 150, and in fact, I had crossed the threshold, 6.7. And this had been, by then, it had been up for probably about 2 months or so. And so it was clearly reproducible because it had shown up twice. And I can tell you when I got this news, this is April 11th of 2011, because after getting this result back, basically I completely changed my diet. I used to have a terrible diet because I didn't think I had anything to worry about. I used to eat desserts all the time. And I completely cut all that stuff out. I haven't had a soda since that day, and I had one bite of wedding cake that didn't taste very good in the summer of 2011. That's the only dessert except for fruit types of things. And I also increased my exercise. So I bike quite a bit, and I doubled my biking, started running. I haven't been running lately. But it took a while. It took about 6 months, and my glucose gradually came down. It's generally stayed down low ever since. Okay, so the important lessons of this study were, first of all, that my genome did, in fact, predict as a risk for disease. And it's only because of that information I, in fact, did follow, especially looking at glucose, and I was able to catch it early. And that's also the case for most diseases. If you catch them early, you can manage them. But if you catch them late, it's very difficult to reverse things. By the time symptoms arise, it's a problem. And so in my case, the genomic information was very helpful. I caught it early. Mine was manageable. I didn't have to take any of the leading drugs, although if I did, I would know what dose to take from my genome as well, because I know I'm going to increase sensitivity for metformin, the leading drug for type 2 diabetes. So my genome could help me there. I hope I don't need that, but I have that information if I ever do. The other thing that was quite remarkable about this study is that when all this occurred, it was right after this nasty RSV infection, respiratory syncytial virus. Again, not very common in adults. I was out for several days. I wish I knew exactly what strain it was. We didn't get that kind of information. But the point is that I did get this, and so we're suggesting it's raising a hypothesis that viral infections could be linked to type 2 diabetes. They have been shown to be linked to type 1 diabetes, but not type 2. Stress responses have been shown to be associated with type 2. So our hypothesis is that my genome has me predisposed to type 2 diabetes, and then the viral infection triggered it. Genome plus environment, right, triggers the disease. It leads to an obvious hypothesis that we're following in other studies, but since we published this, I can tell you about a little over a year ago, once a week I get someone who sends me an anecdotal story with a very similar situation. So again, it's not solid science, don't get me wrong, but there are many other anecdotal stories just like mine where someone, either an individual or someone in their family, often thin like me, got some nasty viral infection and then actually became diabetic. My other bias out of all this is I think diabetes, type 2 diabetes, probably 100 diseases, not one, and we really need to stratify this, and so we're doing lots of studies to get at that. I've gotten pretty late, but I'll run through a few other stories. Because we did all this other omics profiles, we could see the molecular changes occurring to me at a level no one's ever seen before. And I think this will ultimately be very, very powerful for personalized medicine. So my cytokines, again, which are a measure of your immune response, they went ballistic during the viral infections, which we expected. They also spiked at a very mysterious time, about 2 weeks after this RSV infection. It so happens that's a time when other very interesting molecular changes have occurred. I'll probably jump over this, but we did very detailed analysis of my 40,000 components to see what biochemical pathways were changing. And we had to write all kinds of new algorithms to be able to analyze this sort of data. It's nobody had ever done a study like this before. So we had to set up new ways to be able to systematically pull out the data in an unbiased fashion to see what was going on. We don't want to prejudice ourselves. And the bottom line is we can see all kinds of pathways. About 2 weeks after this RSV infection, a spike of all kinds of components, metabolites, proteins, transcripts, RNA, and one of the major pathways that changed was my insulin secretion pathway that's regulated by glucose. We had insulin signaling pathways that spiked down during that time that we could pick out as well. You could actually see biochemical events that were very correlated with things that were relevant going on to me, meaning my glucose misregulation. And we could see these pathways changing at a level no one's ever done before. And we see other kinds of biochemical pathways that are changing too. I don't know how to interpret them yet. And this is why I'd love to do 1 million of these profiles, because quite frankly, when we have 1 million of these profiles and we're summing through the data, we'll see what kinds of new pathways nobody ever realizes. Right now, I just see, I'll tell you, I see a neuroendocrine pathway changing. What the heck does that mean? I don't know. Maybe that happens in everybody when they get diabetes. We don't even know how often people get diabetes quickly like me. How gradual is it? How often does it spike up like mine in a very rapid response? Nobody's done this sort of study. So this is why I would argue we need very detailed analyses. We just got some funding to launch a very small study where we're going to look at about 50 pre-diabetics and see all the molecular changes and the events that are associated with those when people get diabetes. Some fraction of those will. We see other kinds of changes as well, too.